So now we're going to go to the really fun stuff. We're going to talk about what's coming, right? So we've heard a lot yesterday about preclinical discovery and things on the verge of toxicology and pharmacology studies to get to clinical trials. Well, today is the day where we're really talking about those things that are either very close to approaching clinical trials or in clinical trials, and the data that looks like, and the promise and concern and risks in which we need to think about and contemplate as we make decisions for our family members. So we're going to start with pillar one. And I don't think he needs much of an introduction. He's been part of this community since 2017. But Dr. Jim Wilson, who is coming to us from what used to be called the University of Pennsylvania, which is now GEMMABio. He's the CEO and founder of GEMMABio, where all the gene therapy programs he was working on at the University of Pennsylvania have come into this entity, of which they can more aggressively and quickly advance these programs to the clinic. So he's going to talk to you about where we are with the program GTP-220. Jim.
Thank you, Allison, and it's really great to be back. I give you an update each year. Great to see all of you that have been here in the past, and look forward to meeting those that are new to the meeting. Last year has been a busy year, a lot of blocking and tackling and sort of grunt work to get us to the point where we hope to submit the request to open this trial in Q1 of next year. So what I thought I would do today in anticipation of that is very briefly talk about the preclinical data that we have, which hasn't changed much since last time, but try to put into context the platform that we've deployed for Angelman and what experience do we have in that platform in other clinical trials that could be relevant. And I'd also like to touch at the end to follow up with what Ed said was the first valley of death, and I'm going to comment on the second valley of death that ultimately led me to decide to leave the academy after 33 years and become a CEO of a biotech company. Last thing in the world I probably thought about when I graduated from medical school. So these are our funding by FAST and Heather and Richard and Juliet who've done the work. So we're going to talk about how to deliver vectors to the central nervous system. It's something called sort of biodistribution. Can we get the gene to enough cells, and how are we going to do that? The holy grail is to inject the gene delivery vector into the blood to cross the blood-brain barrier. And there may be technologies to do that for gene therapy that are early, and we need to keep our eye on those technologies that may be relevant for us in years to come. There's also a direct injection into the brain, which has been attempted in many different studies. But what we're talking about here, the community has gained some experience with ASOs in injecting the gene delivery vehicle into the cerebral spinal fluid. For ASOs, it's in the lumbar space. But as we tried to leverage that experience for vectors, we did not get adequate distribution to the brain. So it's simply a lumbar puncture closer to the brain in something called the cisterna magna. And this is a view of how the injection would occur, and I'll talk a little bit more about the device and sort of the experience that we have. But studies in non-human primates where we inject a vector very similar, if not identical, to what we'll use in the clinical trial, into this space, and we label it so that we could image where it goes. And this is a study done by Ron Crystal where we imaged the distribution of this vector throughout a non-human primate in an hour, 24, 48, and 72 hours. Wherever there's black, that's where the vector's going, and north is where the brain is, and then south is where the spinal cord is. So you can see that this does widely distribute in the compartments where we want it to. The question is, how does it get into the brain, and how does it distribute to brain cells when injected into the fluid, the cerebral spinal fluid, which does widely circulate throughout the central nervous system. And this is a schematic, and if you look at the vessels that merge into the brain from this space, that the fluid that we inject the vector into tracks all the vessels. So you get wide distribution down into the brain, and then around those vessels you get further distribution, as shown when we look in deep structures of the brain in a monkey, where we can detect a cell that's taken the vector up because it turns green. But also, we've learned that the vector targets cells not only of the brain but the spinal cord. And if you're a patient that has a disease that emanates from the spinal cord, this is great news, but it's something that we also have to be aware of, because we don't believe necessarily we need the same kind of gene transfer efficiency in the spinal cord. And there are two types of cells that the vector gets into. One is the motor neurons, the cell that's diseased and defective in the diseased spinal muscular atrophy, and then the sensory neurons, which are impacted on a number of other diseases called sensory motor neuropathies. So it's really this approach and this kind of distribution that we've leveraged in a wide variety of central nervous system diseases that are in the clinic, and I want to share with you both the safety and the efficacy data, because we're directly leveraging that not only scientifically but from a regulatory standpoint as we think about moving forward. So what we believe is important eventually is not to think about individual programs, but to think about a collection of programs in a platform. We call that a therapeutic platform, where all we do is we change the gene. We don't change the delivery vehicle, we don't change the route of administration, we don't change the manufacturing. And why that's helpful is that if we can simplify and minimize the variables between different diseases, that will allow us to use data from one disease to inform the next disease to determine whether it's going to work and whether it's going to be safe. From a health authority standpoint, it enables you to substantially diminish the timeline and the cost of moving into follow-on diseases. And this is the platform that we decided to deploy for Angelman syndrome a few years ago. And so this is being developed in not only our lab, now our company, and the many companies that we partnered and we started in a variety of diseases called lysosomal storage diseases, adult neurodegenerative diseases, and neurodevelopmental diseases, AS. So the injection, because it's different than a lumbar puncture, although it's the same compartment, it's the same kind of approach, but one needs to exercise additional care when you inject the vector closer to the brain than down in the back. And the vector and the needle goes into that space like in a lumbar puncture. But a colleague of mine at Penn, Brian Buchanan, 8 years ago, when I approached him about translating this into the clinic, has become the world's expert on this as a consultant to FAST. And he basically has trained the entire world on how to do this safely by bringing operators from around the world to Penn, bring them into the interventional radiology suite. We have human cadavers and also non-human primates where we proctor doctors, and any doctor that would be involved in this study would have trained from Brian. He now supports over 44 physicians on behalf of 13 companies, and importantly, this approach has been used to deliver gene therapy vector in a wide range of patients, ranging from 6-month-old patients to 64-year-old patients. The device isn't complicated. It's a syringe, a tube, and a needle. What's important is the skill of the operator to make sure that this is done properly. And what this means is that the vector and the gene therapy will be delivered not at any hospital, but a limited number of hospitals that have adequate training. And these are the sites that are now trained in the United States, Europe, and Israel, and in Brazil. And we're expanding this into other regions of the world through our clinical trials. So I want to give you a brief summary of where this platform that we're going to use in 25 for Angelman syndrome has been used in other diseases, and what are the data so that you know how we think that would inform the potential efficacy and safety of this product for Angelman. What we have in this table, and I know it's busy, but we have the diseases, 1, 2, 3, 4, 5, 6 diseases that are in clinical trials. The first 5 are lethal infantile neurodegenerative genetic diseases due to a defect in a particular gene. And each of these genes encodes a protein that can be secreted from the cell and can benefit an adjacent cell, much in the same way that we think could be relevant in Angelman syndrome, but a little less experience with that. The lifespan for these patients, as I'm going to show you, is literally in years because it's a rapidly degenerative disease. But we progressed these studies through the first 6, and that one is in a BLA submission, which means we've completed all the studies. It's now in front of, or will be in front of FDA for approval. Another one is very close, and the rest are in phase 1-2 studies. The final study, which is gaining some momentum, is using the same approach for an adult disease that causes later onset dementia. So the experience early on with the platform will inform safety, will inform did we get enough gene in, but because the products of these genes are secreted, we could measure non-invasively the product, and we can ask the question how durable it is, because there's the question about sort of, is this a one and done, which is a word then description that I think may overestimate how long we think this will work. But gene therapy, as opposed to traditional therapeutics, should last for a long period of time. Once the gene is delivered to the cell is programmed to express the gene. But until you do the experiment in humans, you don't know. So one thing about all of these diseases that I mentioned, there's something called a biomarker, which you may have heard about yesterday, and that is a measure that you could, an assay that you could conduct in a blood sample or some other cerebrospinal fluid that is abnormal in disease, and and when we correct the disease, that would be corrected. Now that's easier to measure than it is a clinical effect like we heard about in the previous talk, because it's a simple assay. But the crux of whether a biomarker would be useful in clinical development is whether there's reasonable expectation that correction of that biomarker would benefit the patient. And if there is, there's a path forward possibly for an approval from FDA without a long or large clinical trial. Now, in many rare diseases, you don't have enough patients to conduct that disease. The diseases that we're sharing with you now are particularly amenable to measuring biomarkers and that correlate with disease. So the most advanced program came from a product that came out of our lab in a company, GTP-220. They dosed 25 subjects. They were primarily younger. Safety profile was great, and down in the lower left you have the biomarker before and the biomarker later. And you can see that it went down. And GEMMABio has feedback from FDA that that's sufficient for an approval, and they are currently submitting the BLA that should be completed next in the spring. Another disease, MPS I, same company, same product, same approach, different gene, has measured this biomarker and the reduction of this biomarker over time, and that is in weeks, where you see beforehand it's elevated and afterwards it goes down, and it's stably down, which means the gene continues to express. Again, there were 9 subjects dosed, no safety considerations, serious adverse events. It shows durability of correction of this biomarker, and FDA has indicated that possibly for an approval based on this biomarker for this disease. The disease that I want to talk about next is a disease that what came out of my lab was licensed to a company called Passage Bio that I founded, and despite really encouraging clinical data, the program was terminated. And it really was based on this disease and many other diseases that I thought were being shelved that I decided to leave the academy and get on the other side and get control of these products so we can move them forward. This is GM1, another severe disease. Children with the most severe form of this disease usually succumb from their disease within 2 to 3 years of age. But there's a biomarker that we hypothesize, if it's elevated, may correlate with severity of disease, and it's a measure of the substrate that the enzyme reacts. So when the enzyme is deficient, substrate is elevated, and that's shown on the left side. And there are 3 forms of the disease: a very severe form, early infantile, late infantile, and juvenile. And this genetic variation provides us with very nice biology to ask the question, if your disease is less severe, is your biomarker any worse or better? So in the early infantile, patients die within 2 years of age, 2 to 3. Severely elevated. The juvenile form, attenuated disease, not elevated. And when you look at the natural history of these same patients, in the early infantile, there's uniform fatality, no survival for all patients over about 3-year period. But the juvenile form, where there isn't an elevation, there isn't any death. And then the intermediate form is really interesting because it's a mixed bag. Those that have elevated levels of the biomarker in our natural history study did not survive, where those that it wasn't elevated survived. So we went to FDA and asked the question, should this be sufficient for an accelerated approval? And we are in discussions with FDA about that. But it really was based on a clinical trial of GM1, when we inject a vector into the cisterna magna, get the gene into the brain. Do we get a reduction in this biomarker, and is it stably reduced? So I'm just going to share with you some data. Turns out we've dosed 11 subjects. It shows you how simple some of these really rare disease trials can be. 4 at a low dose, 4 at a medium dose, and 3 at a high dose. And at the medium dose, what we show on the left is this biomarker in the cerebral spinal fluid before gene therapy and then after gene therapy. And it's remarkable that it's approaching normal, it's all reduced, but importantly, it's stable out to 720 days that we've followed this one patient. Then the question is, what about clinical benefit? And we've just simply looked at survival in this group. Because this is such a uniformly lethal disease, it may be a bit easier to show benefit, because the 2 that have the early infantile form of this disease are surviving, and from what I understand, doing reasonably well, whereas peers that were part of the natural history study were not surviving. And then finally, if we ever move into adult patients, what are the data that the same kind of technology could realize a safe delivery and a efficient expression and durable expression? And this is an adult onset disease, similar to what Bruce Willis has, although this is a genetic form of that due to a defect in progranulin, which also is a secreted protein, so we could measure the gene product over time. And Passage Bio has moved this into the clinic, and now have dosed 5 patients. They all had low levels to begin with. They went up. The shaded area is normal. It actually was above normal, so we're not having a problem with not enough gene transfer, and over a period of a year continues to express, and these patients will be followed over time. Now, the natural history of this disease is more complicated than the early infantile genetic diseases I spoke about, so it's very likely that, and it's possible, and I don't know this for sure, that if we don't have a really great biomarker, this may need to be a more complicated placebo-controlled trial. So getting to Angelman syndrome, and then finally some final comments about the second valley of death. We've been supported by FAST from the beginning. Our team has continued to enjoy this really collaborative effort, especially as we have moved not only from basic science into translational science. It was a perfect coming together of a group that knows the disease, knows the endpoints, knows the KOLs, with a group, which is FAST, with our group that knows gene therapy. And I would say, and maybe I mentioned it last time, but really without question, the most productive and enjoyable collaboration we've had, and we've had a lot of collaborations. That's not to say that we're constantly pushing the timelines, which we need to be pushed because this stuff, when you get into the blocking and tackling and the grunt work, can take longer than it should. So just to remind you, our approach, it's a gene delivery vehicle that expresses a normal version of UBE3A. There are 3 isoforms. We've did experiments in the past where we introduce these into vectors under different promoters, in particular promoters that drive the expression of the gene in cells such as neurons, the synapsin promoter. And we decided through a variety of in vivo studies in a mouse model to select isoform 1. We had enough data in both mouse models to show the potential for benefit, and also in non-human primates, which are the most sensitive readout of safety, that we would proceed forward. And we declared this GTP-220. Appreciate the applause down there for the announcement of 220. There's a young boy here that gave us an applause too, so thank you guys. Just to summarize at a high level the work that was done, because we presented most of the proof of concept work last year, and actually the year before, is we had to move from research in a research vector to formal, what are called IND-enabling studies. I know you've heard about them, where they have to be more carefully controlled, and there needs to be oversight called quality assurance. And we have to use a product that is very similar to the product that we would manufacture in the clinic. So it's a bigger deal, it's a more expensive deal, and it takes more time and more money, but we've completed all those studies, we have all the reports, the data look great, and we're now in the process of assembling those for this submission in Q1 of next year. But this is the kind of experiment that you do. You have a control, you have different doses, you inject the animals, you follow them over time, over many, many, many assays that attempt to assess whether the gene therapy product worked. And then you have to write fairly extensive reports documenting all aspects of this. And there is very little tolerance for any deviation from the experiment. So if it says the vector is injected on day 1, then it's day 1, not day 2 or day 3. And then non-human primates, again, similar rigor, where you need to inject the vector at different doses, follow the animals over time, follow them for clinical manifestations of any toxicity. In the primates, we're not looking at curing Angelman because they do not have Angelman, but we're looking for safety readouts. And just to summarize, and I know our team presented this yesterday, and I presented most of the data in the mouse studies. When we move from research to our IND enabling, we had the same results. There's some, in a number of these assays, there's evidence that we've improved the abnormalities in the mouse model. And then in the non-human primates, I'm going to talk about safety, so that everyone understands at least what I know about what my concerns are of safety and where this product is going to end up. But there's a real question about when you inject the vector this way, will it get to enough cells in the brain to have an effect? Clearly in a mouse, that's the case. The question is, in non-human primates, are we going to achieve sufficient levels of efficacy? We can't test that directly in the primate model because they do not have Angelman syndrome, but we can determine where the gene goes and how many cells is expressing it, and is it penetrating throughout the brain. And on the top, this is an assessment of the gene-derived expression across many portions of the brain. And then also, when you try to detect expression in the brain using a stain that can detect cells that express the gene, we can detect it as well. The real question is, are we going to get gene distribution in enough cells? If in fact the product of the gene is secreted from the cell that we get the gene into and can benefit adjacent cells in a bystander effect, this then becomes the same kind of disease of the 6 programs that I described to you, all that had really remarkable success. And we believe that is the case. If that's not the case, it's still possible this may be beneficial, but it really relies on how many cells actually harbor the gene. And this is something we can talk about in the panel, but there's a lot of misunderstanding about biodistribution of AAV vectors that I'd be more than happy to spend more time talking about. But if we didn't think that there was a plausible chance that this was going to benefit, we would not be doing this study. So safety. The only safety readout that we've seen in all of those studies in primates and in humans has been evidence when you look at the tissues that there may be some inflammation in a certain type of cell. We've never seen, or actually very, very rarely have we seen any evidence that this causes any problems with these sensory neurons. And it was only in non-human primates that received a very high dose of vector that was a foreign protein. But I still think it's important for you to know what the data are. So what are we talking about? Remember I said when we put the vector in the cerebral spinal fluid, it goes to the brain, but the sensory neurons and the motor neurons, it's the sensory neurons that really take this vector up. And there was evidence in our early non-human primate studies that supported all those clinical trials that when we look carefully at histology of those tissues, and we didn't know it was going to occur, when you do that experiment, you look at all tissues and you determine if you see anything, that there was evidence in the spinal nerves that come out of the spinal cord for a low number, single-digit number of sensory neurons that could have inflammation. And we can show different ways why that's the case. So this was determined actually in the non-human primate toxicology studies for our first clinical trial years ago, MPS I. And our group has done an extensive number of studies. Actually, we've studied this in 541 monkeys through various products to make sure we dialed it in. And there are ways to determine if that has occurred non-invasively, which is you can actually do a study on the skin to assess whether there is any abnormality in the nerve, which requires a procedure. But Juliette Hordeau also developed a biomarker, an excellent biomarker, that we can measure in the blood that would suggest there may be toxicity. Obviously, we're going to follow the patients, the subjects, for any evidence of inflammation of the nerves. We've never seen it, but abundance of caution, we're going to monitor it. And so for GTP-220, we've done the monkey studies, we did the histology, we characterized the level of this damage or inflammation in the neurons, and this is just a summary of hundreds of monkeys through many other programs for evidence that there is a low level of this damage. Again, non-clinical. This is a meta-analysis as background. Wherever something's above 1, this is minimal to moderate. Fortunately, when we did the experiment with GTP-220, there was no evidence in any of the animals of any damage to the sensory neurons. This is the cleanest product out of the many products that we've developed so far. And then just to confirm that, which is what we would do in the trial, rather depend on tissue analysis, measure in the blood a biomarker for this finding. And on the left, we have an example of another product that we've developed, which is typical of low-level sensory neuron pathology, where we see that about day 28 after we inject the vector, the level of the blood goes up, and then it comes down because it's a self-limited process. But for GTP-220, pretty darn flat. So, you know, the translation from monkeys to humans, it's still a science, but I can say, based on 541 monkeys with many, many clinical programs, in the grand spectrum of the product, this is the safest that we've seen in preclinical studies. So I'm going to talk about the second valley of death, just briefly, if you can bear with me. And let's dream of a world, and I don't think we have to dream too much, but we'll dream when we succeed. And we get over the first valley of death and fast help support this, and we get into clinical trials and it's approved. Well, the reality is, as Ed had mentioned, companies are under enormous pressure to come up with a business model as to why to invest in this and get a return on their investment. The solution is charge a lot of money for this injection. And so of the seven products that have been approved, AAV products, one and done products, the cost of these injections that is being paid out in higher income countries ranges from 2.9, can't see over there, but $4.3 million a treatment for the treatment. So it's very likely if you live in a high income country that you'll get access to this product. But the problem is, if you don't live in a high income country, you may not get access to it, because your government payer or your health system that pays for your products will, for the treatments, will not be able to afford it. So what we've proposed is something called Project Hope, where we're building an international coalition of stakeholders for the approved products to find ways to provide access to patients that don't have access to them simply because they live in countries that can't afford them. And that's really why I started GEMMABio. It was, in many of the diseases I've been involved with, we got over the first valley of death, but we have the second valley of death, which is to develop these products and provide global access. And we've been able to start. We've been able to start in Latin America, and a lot of people are skeptical of my plans and my goals and my dreams. Don't underestimate us. We actually were able to convince the Ministry of Health in Brazil to fund our program $100 million so we can move our 6 gene therapy programs forward in the brain. And in return, we're going to provide access in Brazil for a substantially reduced cost when those products are approved. The company can still make money, but that doesn't mean that you can't develop a business plan in a way in which those patients that happen to live in a country that is less affluent than others shouldn't have the same access. This is aspirational and maybe audacious, and I'm going to invite this community to join this effort as we move these programs forward, and assure that as you continue to succeed, that you all benefit from this. So I want to thank you very much.
Excellent talk as always, Jim. Thank you. And I think we all share that vision and that mission, in that, you know, we all have been aspirational from the day our children got diagnosed, and we were told that we were being too aspirational. But don't underestimate us. I think that's the true statement that you said. Don't underestimate us. We will get there.